Life is often described as a combination of material and energy. As such, all organisms have to have some mechanism to obtain both of these. We obtain material by consuming it as food and assimilating it into our biomass. However, we also have to power our metabolism, and our consumption of organic matter is tied to this as well. We react most of the organic carbon we consume with oxygen, essentially oxidizing it, producing carbon dioxide and water. To accomplish this, complex biomolecules are broken down into acetyl-CoA, where it is fed into the TCA cycle in our mitochondria. Acetyl-CoA is reacted with the 4-carbon molecule oxaloacetate to form citric acid, also known as tricarboxylic acid, hence the name tricarboxylic acid or TCA cycle. Through a series of reactions, electron carriers within the mitochondrial matrix take electrons off the molecule, causing the carboxyl groups to be released as carbon dioxide. Each turn of the TCA cycle releases 2 carbon dioxide molecules, and since the acetyl group fed into the cycle has 2 carbons, the 4-carbon oxaloacetate is regenerated after each turn. The reduced electron carriers, NADH and FADH2, then bring the electrons to the electron transport chain in the inner mitochondrial membrane. There, dehydrogenase enzymes oxidize the carriers, and the electrons flow through various carriers within the membrane, such as quinones. As the electrons move through protein complexes known as cytochrome oxidases, the energy of their flow is used to pump protons from the low concentration in the mitochondrial matrix to a high concentration in the intermembrane space. At the end of the electron transport chain, the electrons go to an oxygen molecule, producing water. The flow of protons from a high concentration to a low one drives the ATP synthase enzyme complex, regenerating ATP, the energy currency of the cell, from ADP and orthophosphate. Ultimately, what drives our energy metabolism is redox chemistry. Various elements have differences in electronegativity, and oxygen is more electronegative than carbon. When a more electronegative element oxidizes a less electronegative one, energy will be produced. Why is this? Well, let's take a theoretical element we'll call X, which is less electronegative, and another Y, which is more electronegative. It takes 3 energy units to strip an electron off element X, and 5 units to take it off element Y. But, because of the law of conservation of energy, if element X gained an electron, it would gain 3 energy units, and if element Y did, it would obtain 5 energy units. So, taking an electron off element X will cost 3 energy units, while element Y, getting it, would give 5 energy units. So this redox reaction will give you 2 net energy units. Basically, the larger the difference between the electronegativity of the 2 elements in a redox reaction, the more energy the reaction will produce. Those 3 energy units needed to get the electrons off element X can be seen as the quote-unquote activation energy of the reaction. The utilization of organic carbon for biomass assimilation makes us heterotrophs, and the use of oxygen as the terminal electron acceptor makes us aerobic. Many bacteria are aerobic heterotrophs, and these are responsible for decomposing dead biomass in the environment. Virtually all of these use the TCA cycle in a membrane-bound electron transport chain. In fact, our mitochondria evolved from an aerobic bacteria. Although some of these don't possess a TCA cycle and are thus incapable of completely oxidizing organic carbon. Instead, they'll have an enzyme in their inner membrane which takes a few electrons off an organic molecule, only partially oxidizing it, and passing the electrons down the electron transport chain to an oxygen while excreting the resulting organic molecule. In order to break down larger chunks of dead biomass, known as particulate organic carbon, many prokaryotes involved in decomposition secrete exogenous enzymes. The resulting smaller molecules can be absorbed through the membrane of the bacteria. While the carbon in the biomolecules is completely oxidized and released as carbon dioxide, the other biophilic elements such as nitrogen and sulfur are released as ammonium and sulfides. At the other end of the spectrum, we have methanotrophs, prokaryotes which oxidize methane as an energy source and use it as a carbon source. To do this, they first oxidize methane to methanol and then to formaldehyde, with electron carriers delivering the electrons to the electron transport chain, where oxygen is again used as the terminal electron acceptor. At this point, the carbon has the same general elemental stoichiometry as biomass, CnH2nOn, and thus it can either be oxidized further to power the organism's energy metabolism, or it can be assimilated. If it is oxidized further, it will be converted to a formate and then finally a carbon dioxide. If the carbon is assimilated, the formaldehyde will be fed into one of two pathways. Type 1 and 10 methanotrophs use the RUMP cycle, while type 2 will use the serine cycle. In the RUMP cycle, 3-formaldehyde are reacted with 3,5-carbon ribulose 5-phosphate molecules to form 3,6-carbon hexylose 6-phosphate molecules. 2 ATP molecules are then reacted with 2 of the hexylose 6-phosphates to form 4 3-carbon glyceraldehyde 3-phosphates. One of these is dephosphorylated to become pyruvate and fed into biosynthesis pathways, while the other 3 are reacted with the hexylose 6-phosphate to regenerate the 3 ribulose 5-phosphates for the next turn of the cycle. In the serine cycle, a formaldehyde is reacted with a cofactor known as tetrahydrofolate and reduced to a methylene group. It is then reacted with glycine to form serine. Through a series of reactions, a carbon dioxide is added and malate is reacted with coenzyme A to form glyoxylate and acetyl-CoA. The acetyl-CoA is fed into biosynthesis pathways, while the glyoxylate is converted into glycine for the next turn of the cycle. Methanotrophs can also be given methanol and formaldehyde as carbon sources, and can use these along with formate as electron donors in their energy metabolism. Most heterotrophic organisms uptake other biophilic elements such as nitrogen, phosphorus, and sulfur in a form bound to the biomolecules which they consume. However, since methanotrophs use methane as their sole carbon source, the only other biophilic element bound to it is hydrogen. As such, they have to obtain sulfur, nitrogen, and phosphorus in an inorganic form. Most methanotrophs exist at the interface between oxidizing and reducing environments. This gives them access to ammonia and sulfites from the reducing environment, and these can be readily reacted with glutamate to form glutamine and serine to form cysteine, and thus be assimilated. Orthophosphate can be assimilated by reacting it with an ADP in the ATP synthase enzyme to form ATP. However, other heterotrophic prokaryotes don't have access to sufficient quantities of ammonium sulfide to support growth. Many operational taxonomic units, or OTUs, live in low nutrient or oligotrophic environments. These live off dissolved organic carbon, or DOC. While more complex than methane, these molecules are usually composed of a few carbon atoms bound to hydrogen, with an oxygen or two in the mix. They don't have nitrogen or sulfur-based functional groups attached. Oligotrophic systems are almost always very oxidizing, so sulfur and nitrogen in the system will be in the form of nitrate and sulfate. To assimilate these, an assimilatory nitrate reductase will first use ferredoxin as an electron donor to reduce nitrate to nitrite. Then another enzyme, known as assimilatory nitrite reductase, will reduce the nitrite to ammonia, likely using hydroxylamine as an intermediate, to be assimilated. The sulfate assimilation pathway is a bit different. First, sulfate is reacted with ATP by the ATP sulfurylase enzyme, forming adenosine 5-phosphosulfate, or APS, and releasing the diphosphate group originally bound to the ATP. Then, the adenylyl sulfate kinase enzyme reacts another ATP with the APS, adding a phosphate to the third carbon on the ribose, forming PAPS. Next, a PAPS reductase enzyme takes electrons from a thioredoxin to reduce the sulfur group on the PAPS, forming sulfite and releasing the 3,5-adenosine diphosphate. Finally, an assimilatory sulfite reductase takes electrons from either NADPH or ferredoxin to reduce the sulfite to sulfide. Apart from microbes inhabiting highly oligotrophic environments, those living near oil seeps are in a somewhat similar predicament. They have access to copious amounts of organic carbon, but little ammonium or sulfide to go with it, and thus must uptake nitrogen and sulfur as nitrate and sulfate. This brings me to an important principle in evolutionary ecology, that between R and K selection. R-selected organisms have higher reproductive rates, while K-selected ones have longer lifespans. To understand this concept, let's take a look at the logistical growth equation. Here, The rate at which a population changes over time is equal to the population at the given time multiplied by the intrinsic growth rate, R, multiplied by the difference between the carrying capacity and the population, divided by the carrying capacity. If we plot the population over time, initially it will grow fast, but as it approaches the carrying capacity, the growth rate will slow down asymptotically approaching the carrying capacity, giving a sigmoid-shaped curve. Of course, nutrient influx rates into environments can change over time, and with them the carrying capacity will as well. After a sudden influx of nutrients, the carrying capacity will increase, and during this time, eutrophy adapted R-selected organisms with metabolisms geared towards growing as fast as possible will quickly dominate the system. However, once the nutrients are largely used up, oligotrophy adapted K-selected organisms with metabolisms geared toward conserving energy and resources versus rapid reproduction will dominate the microbial community. If you plot the nutrient concentration against the growth rate of R and K selected microorganisms, you'll see two different curves. These curves follow Michaelis-Menten kinetics, where the growth rate is equal to the maximum possible rate multiplied by the nutrient concentration divided by the sum of the nutrient concentration and the half saturation constant. The half saturation constant is the nutrient concentration that will result in the growth rate half that of the maximum growth rate. Usually, there is going to be a positive correlation between the Vmax and Km, with R-selected organisms having large Vmaxs and Kms, while those of K-selected organisms are very low. So if we plot these on a graph, we can see the R-selected organisms will have the edge with high nutrient concentrations, while K-selected ones will do comparatively better at low nutrient concentrations. Not only that, but the high nutrient concentrations which allow R-selected organisms to flourish are often toxic to K-selected ones. As such, R-selected organisms usually go dormant during oligotrophic conditions, while K-selected ones go dormant under eutrophic conditions. This brings me to another principle of microbial ecology, that being auxotrophy versus prototrophy. Prototrophic organisms can synthesize all the metabolites that they need, such as amino acids and enzyme cofactors, what we call vitamins. We famously can synthesize the essential amino acids, along with many vitamins such as vitamin C and B12, hence we are auxotrophic. R-selected organisms will more often be auxotrophic, since they live in a diverse community of other microbial OTUs, producing the compounds that they need for growth. This isn't the case for K-selected organisms, who live in environments with low population densities, and whatever metabolites other microbes secrete are more likely to be altered by abiotic processes. As such, microbes adapted to oligotrophic habitats are more likely to be prototrophic. While oxygen is seemingly an ideal electron acceptor, given its high electronegativity and abundance in the atmosphere, the presence of oxygen isn't always guaranteed. In stagnant water, the dissolved oxygen will be readily used up by aerobic bacteria, and with low flow rates, it won't be replenished by influx from the atmosphere. As such, microorganisms often have to use other electron acceptors to oxidize organic carbon. This process is called anaerobic respiration. One frequently used variety of alternate electron acceptors are the oxidized nitrogen species, or NOx. The most oxidized form of nitrogen is nitrate, and, as in the aforementioned nitrate assimilation pathway, it is first reduced to nitrite by a nitrate reductase enzyme. However, the respiratory nitrate reductase, or NAR, is of a different variety than the assimilatory nitrate reductase, or NAS. Both of these enzymes have a molybdopterin cofactor at their active site. This nitrite is again reduced further by a nitrite reductase, but whereas the assimilatory version has a multi-heme functional group at its active site, the respiratory nitrite reductase has a copper ion and reduces nitrite to nitric oxide, arguably the most reactive nitrogen intermediate. The nitric oxide is then reduced to nitrous oxide by nitric oxide reductase, or NOR. Nitrous oxide is a gas, so some of it will diffuse out of the system. Finally, the nitrous oxide reductase enzyme, or NOS, reduces the nitrous oxide to molecular nitrogen, where it goes up into the air with a pull of atmospheric nitrogen. This whole process is called denitrification and can be viewed as a sink for bioavailable nitrogen. While some microorganisms possess all of the aforementioned enzymes and can thus produce molecular nitrogen from nitrate, many can only carry out a few steps, meaning that they have a truncated denitrification pathway. In these scenarios, various microbial OTUs in the environment quote unquote pass intermediates between each other, so the reduction of nitrate to nitrogen is a quote unquote community effort. Now, how do denitrifying organisms fare when oxygen is present? Not very well. Since oxygen is more electronegative than nitrogen, aerobic organisms can get more energy per gram of organic carbon metabolized, allowing them to grow and reproduce faster and thus outcompete denitrifiers. However, it should be noted that the enzymes which carry out denitrification are fairly similar to those employed in aerobic metabolism, and a large fraction of aerobic prokaryotes, and even some fungi for that matter, can facultatively utilize denitrification under oxygen-limiting conditions. Another form of nitrogen-based respiration is known as dissimilatory nitrate reduction to ammonia, or DNRA. As its name suggests, this pathway involves reducing nitrate to ammonia. This will produce less energy per mole of carbon oxidized than denitrification, but it involves the transfer of more electrons, 8 versus 5, so it may be more energy efficient, but this is yet to be determined. It's not currently known why some microbes employ DNRA over denitrification, but some features of environments where it is prevalent include high organic carbon loads with high to C to N ratios, high rates of sulfate reduction, and high summer temperatures. DNRA is initiated by a third variety of nitrate reductase, known as the periplasmic nitrate reductase, or NAP, which again produces nitrite. The nitrite is then reduced to ammonia by the cytochrome C nitrite reductase, which has more similarities to the assimilatory nitrate reductase than it does the respiratory version due to its heme cofactors. Most of the NOx used in nitrogen respiration was originally bound in organic matter, but this isn't the case for the next major alternative electron acceptor, iron, whose abundance is largely dependent on the local geochemistry. Now, you might be thinking, wait, how could iron be used as an electron acceptor if it's less electronegative than carbon? Well, that's true, but when we're talking about how oxidized the carbon is, it's with regards to an unequal sharing of the electrons. Since carbon is more electronegative than hydrogen, when it's covalently bound to it, the shared electrons will tend to be closer to the carbon atom, making it reduced. Meanwhile, when it's bound to an oxygen atom, which is more electronegative, the electrons will spend less time around the carbon, making it oxidized. However, the iron used in iron respiration will be an iron 3 ion. It's not that the iron is unequally sharing electrons, it's already completely lost 3, which makes electrons more attracted to it, i.e. effectively more electronegative, allowing it to act as an electron acceptor. The iron 3 is reduced to iron 2, but it can't be further reduced to zero valent or metallic iron. One challenge iron respiring organisms face is the low solubility of iron oxides, or rust, their electron acceptor. Usually, these microbes will employ one of two strategies. One is to use chelating agents to clip bits of iron oxides off a particle and bring it into their cell. Another is to put the enzymes which reduce the iron oxides on the outer membrane. Here, the microbes bind to an iron oxide particle and pass the electrons to it directly. Apart from iron, many other metals have been documented being used in anaerobic respiration, including manganese, vanadium, chromium, molybdenum, cobalt, palladium, uranium, arsenic, gold, and mercury. While many microorganisms are obligate iron reducers, no one has yet confirmed the existence of microbial OTUs which only carry out metabolism by respiring these other metals. This is likely due in part to the abundance of iron compared to the other metals. Most often, the oxidized form is less soluble in water, but this isn't always the case. For uranium, the reduced form is insoluble. Iron is further down the redox ladder than nitrate, so dissimilatory iron reducers are usually outcompeted by denitrifiers for the same reason that denitrifiers are almost always outcompeted by aerobic organisms. But some of these metal ions, like cobalt 3, have an even greater redox potential than diatomic oxygen. While facultative iron respiration is utilized by some aerobic microorganisms in non-altered habitats, it isn't as common as facultative denitrification. Also, aerobes and denitrifiers are known to conduct methanotrophy, but no metal respirers are yet known to do this. A major difference between aerobic and anaerobic prokaryotes is that aerobes use the TCA cycle to completely oxidize carbon with a wide variety of pathways that break down organic matter and bind it to coenzyme A, forming acetyl-CoA to be funneled into the cycle. Conversely, anaerobic respirers will just have an assortment of enzymes which partially oxidize certain organic molecules and pass the electrons down the ETC to an inorganic electron acceptor. Thus, individual OTUs will not be able to completely oxidize organic carbon. They may be able to produce carbon dioxide, but that's because the functional group oxidized is a carboxyl group. So, in anoxic environments, the complete oxidation of biomass is generally another community effort. If we go down the next rung of the redox ladder, we come to the least electronegative electron acceptor used by heterotrophs, sulfur. The most commonly used chemical in sulfur respiration is sulfate, the most oxidized form of sulfur. In a manner similar to assimilatory sulfate reduction, the sulfate respiration pathway is again initiated by the sulfate adenyltransferase, which reacts the sulfate with ATP to form APS. However, after this, the two pathways diverge. In the respiratory path, an adenyl sulfate reductase takes electrons from a thioredoxin to reduce the sulfur group to sulfite, releasing the adenosine monophosphate. The sulfite is then reduced to hydrogen sulfide via the dissimilatory sulfite reductase enzyme, or DSA. However, sulfa is available in many forms in the environment, including elemental sulfa, thiosulfate, tetrathionate, and dimethyl sulfoxide, or DMSO. These are metabolized by enzymes in the aforementioned molybdotierin oxidoreductase enzyme family. Tetrathionate is reduced to 2-thiosulfates by tetrathionate reductase, or TTA, which is then reduced to sulfite and hydrogen sulfide by the thiosulfate reductase enzyme, or PHS. The sulfite is finally reduced to hydrogen sulfide by the DSA. DMSO is reduced to dimethyl sulfide by DMSO reductase, and while these organisms cannot reduce elemental sulfur directly, a sulfide will react with elemental sulfur. This breaks the ring and forms a polysulfide chain. The polysulfide reductase enzyme, or PSA, will reduce the sulfur atom at the end of the chain, one at a time, releasing a hydrogen sulfide until every atom is reduced. While aerobic organisms use quinones as intramembrane electron carriers, sulfur-reducing microbes use menaquinones. Sulfate reduction is also largely dependent on the local geochemistry and is very prevalent in the oceans. Sulfate is the third most abundant ion in the oceans after sodium and chlorine, and as a result, about 50% of all remineralization in the oceans is carried out by sulfur-reducing microorganisms. Even though they're outcompeted by aerobic organisms when part of a microbial community, when in pure cultures, most denitrifiers and dissimilatory metal-reducing prokaryotes can grow in the presence of oxygen, making them aerotolerant anaerobes. This isn't the case for the majority of sulfate-reducing microbes, whose growth is inhibited by the presence of oxygen, making them strict anaerobes. That said, a select few aerobic organisms which facultatively utilize sulfur respiration have been documented, but they occurred in habitats subjected to anthropogenic perturbations, which induced redox dynamics that wouldn't occur otherwise. Some sulfur-reducing microbes are methanotrophs as well. But what happens when no electron acceptors are available? Instead of oxidizing or burning organic matter, microbes can break it down. The process by which biomolecules are broken down into simpler organic ones can be described as fermentation. Sometimes, in our own body tissues, we can't deliver oxygen fast enough to meet our energy demand, so our cells employ this strategy. Glycolysis is the main conduit by which glucose is broken down into acetyl-CoA and fed into the TCA cycle. As it is broken down, one molecule of glucose will produce two net ATPs and is partially oxidized by NAD+. The end product of glycolysis is pyruvate, which is reacted with coenzyme A to produce acetyl-CoA, releasing the N-carboxyl group as carbon dioxide. Meanwhile, the resulting NADH will bring the electrons to the mitochondrial electron transport chain. However, without oxygen, the TCA cycle and electron transport chain shut down, so the pyruvate isn't converted into acetyl-CoA. Instead, the NAD+ are regenerated when NADH reduces it to lactate. This allows glycolysis to keep proceeding and replenishing ATP, producing lactate as the end product. Apart from the production of lactate, there are many other fermentation pathways utilized by microorganisms. Too many to be covered in this video. But one thing to note is that quite a few microbes will react the pyruvate with coenzyme A. This may seem similar to what occurs in respiration, with the key difference being that the binding of coenzyme A isn't coupled with the reduction of NAD+. So, while in a respiratory pathway, the carboxyl group on the pyruvate is released as a carbon dioxide, in a fermentative pathway, it's released as a formate. The formate is then disproportionated into hydrogen and carbon dioxide by a formate dehydrogenase enzyme complex. As for the acetyl-CoA, one of two things could happen. The acetyl-CoA can be reacted with NADH to replenish NAD+, releasing the coenzyme A and resulting in the production of ethanol. Alternatively, it can be reacted with the phosphate to form acetyl-P, which is then reacted with an ADP to produce ATP, and acetate is the final product, which is then excreted. Which strategy is employed will often depend on the redox balance of the cytoplasm. If large quantities of NADH are being used by other cellular processes, there will be less need for the cell to regenerate it via fermentation to keep glycolysis going. So, it would be more inclined to utilize acetate fermentation. But if it couldn't replenish sufficient quantities of NAD+, the microbe would probably employ ethanol fermentation. Instead of an ATP synthase enzyme, the ATP regenerated by fermentation is produced via direct interactions with the molecules being fermented. This process is known as substrate-level phosphorylation, and it will only produce 1/16 the energy per mole of glucose metabolized that aerobic respiration will. Although some microorganisms only produce energy via fermentation, many, if not most, aerobic microorganisms can facultatively carry out fermentation should conditions become anoxic to support their energy needs, although most cannot sustain themselves off fermentation indefinitely. Many of the microorganisms which inhabit plant roots are obligate fermenters and carry out diazotrophy. This is where nitrogen is taken out of the atmosphere and assimilated into an organism's biomass. This is accomplished by the nitrogenase enzyme, which reduces it to ammonia to be reacted with glutamate to form glutamine. The diatomic nitrogen molecule is held together by a triple bond, which is very hard to break. The nitrogenase enzyme has a very complex iron molybdenum cofactor at its active site and takes 16 ATPs per nitrogen molecule split. Having these diazotrophic symbionts in their roots allows some plants to grow in soils with very low nitrogen content. As I mentioned before, many eukaryotes, including us, facultatively use fermentation, but some unicellular eukaryotes like Giardia, and possibly a parasitic worm, power their energy metabolism solely by fermentation. So, fermenters break complex biomolecules into two carbon molecules like acetate and ethanol, but can they be broken down even further? The answer is yes, and this is done by a group of microbes known as methanogens. As their name suggests, methanogens produce methane, and one variety known as acetoclastic methanogens metabolize acetate. They disproportionate or split the molecule into methane and carbon dioxide. While this involves breaking down organic carbon, there is disagreement as to whether or not acetoclastic methanogenesis should be called fermentation. This is because it doesn't produce ATP via substrate-level phosphorylation. Instead, as electrons flow from the carboxyl group on the acetate to the methyl group, they pump protons across a membrane against a concentration gradient, which is then used to power ATP synthase. Another type of methanogens are known as methylotropic methanogens, who obtain energy by oxidizing the carboxyl group on certain organic molecules and using them to reduce a methyl group on another organic molecule. These are often simple sulfur and nitrogen-based compounds, like dimethyl sulfide and trimethylamine. Most methanogens are extremely sensitive to oxidative stress, being inhibited by sulfate, let alone oxygen. But recent research has shown that methylotropic methanogens, which live in sandy sediments and metabolize DOC released by algae, are more aerotolerant than we thought was possible for methanogens to be. Now that we've covered the major heterotrophic metabolisms, we can switch gears and talk about autotrophic and lithotrophic organisms. Autotrophic organisms obtain carbon in an inorganic form, either carbon dioxide or carbonate ions, and lithotrophic organisms power their metabolism by the oxidation and reduction of inorganic compounds. Here, we'll start at the bottom of the redox ladder with carbon and hydrogen. While carbon can be oxidized by a wide variety of other elements, it is more electronegative than hydrogen, so it can be used as an electron acceptor to oxidize hydrogen. One variety of microorganisms which take advantage of this are the acetogens. These organisms utilize the Wood-Young-Dahl pathway for both carbon assimilation and energy metabolism. This pathway has two main branches, the methyl and carbonyl branch. The methyl branch starts with a hydrogenase enzyme, which takes electrons off a hydrogen molecule and then passes them to a formate dehydrogenase enzyme, which reduces a carbon dioxide molecule to formate. The formate is then reacted with ATP to form formyl P, and this is then reacted with tetrahydrofolate, or THF, to become formyl THF, releasing the phosphate group. The formyl group is then incrementally reduced to a methyl group by methylene THF cyclohydrolase, dehydrogenase, and reductase enzymes, using NADH as the electron donor. On the carbonyl branch, carbon dioxide is reduced to carbon monoxide by the carbon monoxide dehydrogenase acetyl-CoA synthase enzyme complex, which uses ferredoxins as the electron donor. This enzyme complex then reacts the carbon monoxide with methyl THF and coenzyme A to form acetyl-CoA, releasing the THF. From here, one of two things can happen. First, acetyl-CoA can be fed into biosynthesis pathways. Second, and this is what happens about 90% of the time, the acetyl-CoA is reacted with an orthophosphate to form acetyl-P, which is then reacted with ADP to form ATP, replenishing the one used to phosphorylate the formate. So, this pathway uses one ATP and produces one ATP, so there's no net production of ATP. Now, how could it be used for energy metabolism? Well, the electrons that reduce the formyl THF ultimately came from hydrogen as well. Another hydrogenase enzyme oxidizes a hydrogen and uses the electrons to reduce a ferredoxin. The ferredoxin then shuttles them to an RNF complex where they flow to an NAD+, which gets reduced to NADH and then reduces the formyl THF. NAD+ is more electronegative than ferredoxin, so the flow of electrons can be used to pump ions, either protons or sodium ions, against a cross-membrane gradient. This gradient is then used to power their ATP synthase enzyme. But while the aerobic respiration chain will have several cytochrome oxidase proteins to pump ions that each electron will flow through, the difference in electronegativity between hydrogen and carbon is much less than that between oxygen and carbon. So, each electron in the acetogenic electron transport chain can only pump ions through a single complex. Apart from carbon dioxide, acetogens can uptake carbon monoxide to be used for carbon assimilation, and formate for assimilation and energy metabolism. Apart from acetogens, a third variety of methanogens, known as hydrogenotrophs, use hydrogen to reduce carbon dioxide. To do this, methanogens use their version of the Wood-Young-Dahl pathway with a few modifications. Instead of forming a formyl P, a hydrogenase takes electrons from hydrogen, which reduces a carbon dioxide and reacts with a methanofuran, or MF, forming formyl MF. This is then reacted with H4-MPT instead of THF to form formyl H4-MPT, releasing the MF. Hydrogenase enzymes will metabolize more hydrogen, but instead of using NADH to shuttle electrons to the formyl H4-MPT to reduce it to a methyl group, another electron carrier known as F420 is used by methanogens. The formyl H4-MPT is reduced to methylene H4-MPT and finally methyl H4-MPT. Here, we come to another bifurcation point. Carbon monoxide dehydrogenase acetyl-CoA synthase can reduce carbon dioxide to carbon monoxide, which will then be reacted with the methyl H4-MPT and coenzyme A, forming acetyl-CoA, which is then fed into biosynthesis pathways, while the H4-MPT is released. If not, the carbon will be continued to be reduced to power the organism's energy metabolism. The methyl group will be transferred from the H4-MPT to coenzyme M. The carbon is bound to coenzyme M via a thioether. The methyl-CoM is then reacted with the coenzyme B, which forms a disulfide bond with coenzyme M, releasing the methyl group as methane. A hydrogenase will then oxidize another hydrogen, and the electrons are then used to reduce the disulfide bond binding coenzyme M and B by the heterodisulfide reductase enzyme. This produces two thiol groups, splitting/regenerating coenzymes M and B, while the methane is released as the waste product. Unlike acetogens, Methanogens are known to use membrane-bound electron carriers like the aforementioned menaquinones and another type known as methanophenazine. These shuttle electrons between the hydrogenase enzymes, ion pumps, and the reducing enzymes. Both acetoclastic and methylotrophic methanogens also use their own version of the Wood-Young-Dahl pathway. While hydrogenotrophic methanogenesis is essentially an extension of the methyl branch, acetoclastic methanogenesis starts as a reversal of the final step of the Wood-Young-Dahl pathway, which flows up the carbonyl branch but flows down the extension of the methyl branch. Meanwhile, methylotrophic methanogens flow the methyl group down the extension of the methyl branch, while the carboxyl group flows up the upstream end of this branch. While most of the metabolic processes we've discussed so far are present in both bacteria and archaea, all known methanogens are archaeal. Methanogens and acetogens both live in areas where hydrogen is being produced. This can be among masses of decomposing organic matter, where it's being released by fermentation, or it can be geothermal. Volcanic vents such as deep-sea black smokers will release hydrogen, but there's another type of hydrothermal system which doesn't get as much attention known as serpentinizing systems. Whereas in volcanic vents, the water is superheated by coming in contact with magma, and thus getting some volcanic gases dissolved in it. Serpentinizing systems are powered via rocks chemically reacting with the water. Ultramafic minerals like olivine and pyroxene react with water to form serpentine, magnetite, and hydrogen. While volcanic vents are nominally acidic, serpentinizing systems are alkaline with a pH between 9 and 10. This causes most inorganic carbon to be completely deprotonated in the form of carbonate ions, which regularly precipitate out with magnesium and calcium ions, resulting in the systems being very carbon limited. Going up the redox ladder from the electron acceptor side, oxidized sulfur species can also be used to oxidize hydrogen. These chemolithotrophic sulfur reducers use hydrogenase enzymes to oxidize hydrogen, while using the same system sulfur-aspiring organisms use to reduce sulfur. There are two non-homologous varieties of the hydrogenase enzyme, one with an iron-sulfur cluster at the active site and another with an iron-sulfur-nickel cluster. Usually, the iron-sulfur-nickel hydrogenases are used to oxidize hydrogen, while the iron-sulfur variety is used to produce it, but there are many exceptions to this rule. Even if they get energy by using sulfur to oxidize hydrogen, these sulfur-reducing microbes still need to produce organic carbon. Since hydrogen is less electronegative than carbon, it can be readily reacted with carbon dioxide to form organic molecules. Some use the Wood-Young-Dahl pathway to produce acetyl-CoA, but since their energy demands are being met by sulfur reduction, the acetyl-CoA isn't converted to acetate to regenerate ATP and is instead input into biosynthesis pathways. Some iron-reducing microorganisms use hydrogen as an electron donor to power their energy metabolism, but instead of carbon dioxide, they uptake organic carbon in the environment, and rather than oxidizing it, they assimilate it into their biomass. Using the redox chemistry of inorganic compounds while utilizing organic carbon for biosynthesis makes these organisms chemolithoheterotrophs. Many denitrifiers and aerobic microorganisms can also use hydrogen as their electron donor. They again use the enzymes used by denitrifying and aerobic respirers to metabolize their electron acceptor. Hydrogenase enzymes are inhibited by the presence of oxygen, meaning aerobic hydrogenotrophs live in environments with low oxygen concentrations. This makes them microaerophiles, meaning that although they use oxygen, if exposed to dissolved oxygen concentrations in equilibrium with the atmosphere, these bacteria would be poisoned. To assimilate carbon dioxide, most aerobic hydrogenotrophs use the Calvin cycle. Here, a hydrogen is oxidized and the electrons are used to reduce NADP to NADPH. NADPH is an NADH with an extra phosphate group. In the cytoplasm, 3 carbon dioxide molecules are reacted with 3 5-carbon ribulose 5-biphosphates, or RUBP, to form 3 short-lived 6-carbon intermediates, which quickly break down into 6 3-carbon 3-phosphoglyceric acid molecules. This is then reacted with ATP and reduced by the NADPH to form 6 3-carbon glyceraldehyde 3-phosphate molecules, or G3P. One of these is reacted with another G3P to form glucose, which is then fed into biosynthesis pathways. Meanwhile, the remaining 5G3P are react with ATP to regenerate the 3RUBP for the next turn of the cycle. As we go up the redox ladder from the electron acceptor side, you'll get a greater difference in electronegativity between hydrogen and its oxidant, and thus will get more energy per mole of hydrogen metabolized. Acetogens get 26 kilojoules per mole of hydrogen oxidized, while aerobic hydrogenotrophs get 286, meaning that they have to metabolize less hydrogen per mole of carbon assimilated. Remember, assimilation requires more than carbon reduction. The organic carbon then has to be assembled into biomolecules, a process that requires energy. On the electron donor side, the next rung is carbon. Now, we often think of inorganic carbon as being completely oxidized, but there's also carbon monoxide. Sulfate reducers, denitrifiers, and aerobic microbes are all known to use carbon monoxide as an electron donor to power their energy metabolism. While this pathway is a lithotrophic one, the organisms who utilize it are nominally heterotrophic and use carbon monoxide oxidation facultatively to supplement their energy requirements. Often, these microbes live in soils where UV light interacts with volatile organic compounds to produce carbon monoxide. Carbon monoxide is oxidized to carbon dioxide by the carbon monoxide dehydrogenase enzyme. And to clarify, this enzyme is unrelated to the reductive carbon monoxide dehydrogenase acetyl-CoA synthase enzyme complex. Now we'll look at sulfur oxidation. Either diatomic oxygen or nitrate are used as the electron acceptor. To oxidize the sulfur, one of two pathways are used. The first is the sulfur oxidase or SOX, where one of the enzymes forms a disulfide bond with a hydrogen sulfide molecule, which is then incrementally oxidized to a sulfonic acid, followed by a sulfoxide, and finally a sulfone group, before a final pair of electrons are removed, releasing the sulfur as a sulfate. The other major pathway is the sulfide quinone oxidoreductase or SQR pathway. Here, sulfide is oxidized via quinone in a membrane-bound SQR enzyme and forms elemental sulfur. A rhodinase then oxidizes it to sulfite, and the final steps are catalyzed by the oxidative versions of adenyl sulfate reductase and the sulfate adenyl transferase enzymes used by sulfur-aspiring organisms. In a manner which can be seen as the reversal of the sulfur reduction pathway, the oxidative pathway binds an AMP to the sulfite and produces either an ATP or ADP when the sulfate is released. Tetrathionate and thiosulfate are usually oxidized by modified versions of the SOX pathway, while environmentally occurring elemental sulfur molecules are oxidized by entering the SQR pathway at the second main step. There are two other less frequently used pathways. One is a modification of the SQR pathway, where sulfite is oxidized to sulfate by a sulfite dehydrogenase enzyme. The other has the sulfite oxidized to sulfite by an oxidative version of the DSR enzyme. Essentially, the sulfate reduction pathway reversed. In a manner similar to the hydrogenotrophs, sulfur oxidizing microbes couple the enzymes used to oxidize sulfur to the same ones used by heterotrophic aerobes and denitrifiers to reduce oxygen and nitrogen. However, since hydrogen is less electronegative than carbon, and hence there is a large difference in redox potential between it and oxygen, each electron can pump more protons against the concentration gradient. Since sulfur is more electronegative than carbon, in sulfur oxidation, each electron will pump fewer ions than they would in aerobic respiration. Sulfur oxidizers also usually use the Calvin cycle to assimilate carbon dioxide into their biomass. But wait, you might ask, how do they reduce carbon dioxide if they metabolize sulfur, and sulfur is more electronegative than carbon? Well, as the electrons flow from the sulfide to the oxygen on the main electron transport chain, most of the ions that they pump against the cross-membrane concentration gradient are used to drive the ATP synthase enzymes. However, there is another electron transport chain which links hydrogen sulfide to NADP. But this one has the function reversed. Instead of using the flow of electrons from a less to more electronegative element to pump ions against the concentration gradient, the flow of ions from a high to low concentration are used to drive electrons from the more to less electronegative element. Some of the ions pumped by the main electron transport chain are used to drive this minor one to produce NADPH for the Calvin cycle. These organisms play a diverse set of ecological roles, but perhaps the most iconic one is in deep-sea vents. Here, they react hydrogen sulfide emitted by the vent with oxygen in the water column to drive the production of organic matter, and in so doing form the base of the deep-sea food chain. Many of these microorganisms act as symbionts for tube worms, inhabiting their body tissues and providing them with food. While not as prevalent as they are in hydrothermal systems, sulfur oxidizers play a key role in the global sulfur cycle. When sulfur is bound in organic matter, it will be in the form of a sulfide group. This is released when the organic matter is burned or broken down by heterotrophs. This will then be converted into sulfate by sulfur oxidizing microorganisms. When large quantities of sulfites are suddenly released into the environment, sulfur oxidizers, who are usually a small part of the community, will proliferate rapidly. Those which utilize the SQR pathway will produce granules of the elemental sulfur intermediate, which can be seen as aqueous clouds from the air. As mentioned earlier, sulfate respirers are responsible for roughly half of all remineralization in the world's oceans, but pretty much the entire water column is oxic, and most sulfate reducers are inhibited by the presence of oxygen. This means that in the water column, most dissolved organic matter is oxidized by aerobic bacteria, while most of the particulate organic matter sinks to the seafloor. Here, aerobic microbes in the top layer of sediment quickly consume the oxygen. This leaves the deeper layers anoxic, allowing sulfate reducers to oxidize most of the organic carbon that settles there. At the oxic-anoxic interface of the sediment, sulfur oxidizing microbes use oxygen diffusing from the water column to oxidize the sulfides produced by sulfur-aspiring microorganisms. While living at the oxic-anoxic interface gives sulfur oxidizers access to their electron donor and acceptor, it will have lower levels of oxygen than the oxic layer and lower levels of sulfide than the sulfitic or eosinic zone. One variety of microbes known as cable bacteria have evolved a way to have their cake and eat it too. These organisms grow in a linked series of cells or filaments which extend from the oxic to eosinic zone. On the outer side of the cellular envelope, there are bundles of proteins or cables with large quantities of iron-sulfur clusters to conduct the flow of electrons from sulfides in the more reducing to the more oxidizing environment, where oxygen or nitrate are used as the terminal electron acceptors. This allows cable bacteria to metabolize far more hydrogen sulfide than unicellular prokaryotes, which live at the oxic-eosinic interface, could. Apart from oxidizing or reducing sulfur, some microbes employ a third strategy: disproportionating or fermenting it. Here, elemental sulfur or octasulfur and thiosulfate are broken down into sulfate and hydrogen sulfide via interactions with water. The disproportionation of thiosulfate is an energy-producing reaction on its own, but in order for elemental sulfur disproportionation to be energetically favorable, it has to be tied to the reduction of iron 3 to iron 2. This brings me to the chemotrophic oxidation of iron. This is conducted by iron oxidase enzymes and can be carried out by aerobes or denitrifiers. Like iron-reducing bacteria, most iron oxidizers have their iron metabolizing enzymes on the outer surface of their cell membranes. While the substrate iron oxidizers use, iron 2, is highly soluble in water, the resulting iron oxides precipitate out and would accumulate in the cell if the iron oxidizing enzymes were on the inside of the cell. Because the reduction potential of iron 3 to iron 2 is only slightly less than that of oxygen to water, comparatively little energy can be produced by the oxidation of iron, which means iron oxidizers have to oxidize a lot of iron per mole of carbon assimilated. Some microorganisms also use manganese, and even less energy can be obtained from its oxidation. Perhaps the most iconic role of iron oxidizing organisms is in the corrosion of shipwrecks. Here, metallic iron in the hull reacts with oxygen in the water column to form a thin layer of ferrous iron hydroxide minerals. These are then oxidized to iron 3 by microbes, forming a hematite and resulting in the formation of rusticles like the ones on the wreck of the Titanic. Iron oxidizers are also very prevalent in deep-sea vent communities. The plumes which give black smokers their name are made of iron sulfide precipitates, and these are metabolized by microbes capable of oxidizing both sulfur and iron. Most of the compounds utilized by chemolithotrophs are metastable. This means that while they can be reacted to produce energy, they won't do spontaneously under conditions organisms could survive in. However, this isn't the case with iron. Iron too will readily react with oxygen in the water column, which happens more rapidly under acidic conditions, meaning that iron oxidizing microbes have to compete with abiotic processes for substrates. This is true for both aerobic and denitrifying iron oxidizers, as ferrous iron will also readily react with nitrite in a process known as chemodenitrification. This can account for up to 30 and 40% of all nitrous oxide emissions in estuaries and acid sulfate soils respectively, and proceeds at a greater rate in alkaline conditions in aquatic environments and acidic conditions in terrestrial ones. While nitrite is responsible for the majority of chemodenitrification, in hard water environments with a pH in excess of 9, nitrate will oxidize iron, and nitrous oxide itself will react with iron. Although only 2% of the diatomic nitrogen released by acid sulfate soils is produced by nitrous oxide reacting with iron. Now I move on to the last major chemotropic process, nitrification, or the oxidation of nitrogen. This process starts with ammonification. In a manner similar to the sulfides, nitrogen in organic matter is in the form of an amino group. When heterotrophs remineralize the organic matter, the amino groups are released as ammonia, a process called ammonification. Nitrification is initiated by the ammonium monooxygenase enzyme, which oxidizes ammonia to hydroxylamine. The hydroxylamine oxidoreductase then oxidizes the hydroxylamine to nitrite, and in a manner similar to denitrification, this step involves a nitric oxide intermediate. The nitrite is finally converted into nitrate by the nitrite oxidoreductase. Because nitrogen to sulfur ratios in organic matter are usually around 16:1, nitrifiers are more abundant in most environments than sulfur oxidizers, who metabolize sulfide released by decomposition. However, since carbon to nitrogen ratios in organic matter are usually between 7:1 and 4:1, most often, the nitrate produced by nitrifying bacteria isn't enough to oxidize the organic matter in a system should conditions become anoxic. The main exception to this rule is among the microbes in animal burrows. This is because while animals consume and metabolize organic matter to power their energy metabolism, they can oxidize the carbon but not the nitrogen, and instead excrete it as either ammonia, uric acid, or urea. As a result, the C to N ratios of organic matter in animal burrows are usually between 3:1 and 1:1. This means that once nitrifiers have oxidized all the nitrogen to nitrate, there is often enough nitrate to oxidize the remaining organic matter should conditions become anoxic. As a result, animal burrows are responsible for a disproportionately high quantity of the nitrous oxide emitted in marine habitats. Now, you might think that nitrifiers and aerobic heterotrophs would compete with the animal for dissolved oxygen, but this isn't the case. In fact, fanning activity by the animal irrigates the sediment with oxygenated water, giving the microbes access to more dissolved oxygen than they could get otherwise. The surrounding sediments become anoxic when the animal leaves to forage. Even though most ammonia oxidation uses oxygen as the electron acceptor, one variety, known as anammox, uses nitrite as the electron acceptor, producing diatomic nitrogen. The exact mechanism by which this happens is not fully understood, but it does appear to involve a hydrazine intermediate, which is produced by a hydrazine synthase enzyme and is oxidized further by a hydrazine dehydrogenase. The involvement of either a nitric oxide or nitroxyl intermediate have also been proposed. In bacteria which carry out anammox, this process is performed in a membrane-bound compartment known as an amoxosome. The membrane includes a unique variety of lipids known as laterans. Anammox bacteria are used in wastewater treatment plants to remove nitrates before discharge. This can cut down operating costs by as much as 60%. Now, we're back to the top of the redox ladder with oxygen. So, how can it be oxidized if it's the most electronegative element widely available? Well, more electronegative elements can be used to reduce less electronegative elements, but here's the catch. It requires versus produces energy. The energy to oxidize oxygen in its reduced form, i.e. water, comes from sunlight in a process known as photosynthesis. The electrons stripped from the water are then used to reduce carbon dioxide to organic carbon for biomass assimilation. The oxygen is oxidized by being bound to another oxygen atom, forming diatomic oxygen, which is then released as a waste product. This process is responsible for over 99% of all the production of organic matter on the Earth, as well as all of the oxygen in the atmosphere. One variety of prokaryotes carries out oxygen-producing or oxygenic photosynthesis, which are known as cyanobacteria or blue-green algae. Many other organisms carry out oxygenic photosynthesis, including diatoms, chrysophytes, rhodophytes, euglenids, chlorophytes, charophytes, and most famously land plants. However, these carry out photosynthesis using organelles called chloroplasts, which are the descendants of cyanobacteria engulfed by eukaryotes long ago. So, cyanobacteria could be said to be solely responsible for oxygenic photosynthesis. The photosynthetic machinery is divided into two main components: the light reactions responsible for oxidizing water, and the dark reactions which reduce carbon dioxide. Cyanobacteria have membrane-bound compartments within their cells known as thylakoids, and within the membranes are light-harvesting protein complexes, photosystem I and photosystem II. Within each photosystem are the photosynthetic pigments chlorophyll and carotenoids. Carotenoids are lipid compounds which act as antenna pigments, directing the photons which enter the photosystem toward the reaction center. Here, the light is absorbed by a chlorophyll, a porphyrin-based molecule with a magnesium ion in the center. This is where the electrons are excited or energized to be passed down the electron transport chain. These electrons come not from the photooxidation of oxygen, but of manganese from a manganese-oxygen calcium cluster. This cluster is known as the oxygen evolving complex. Through a series of photon strikes, the manganese 3 is oxidized to manganese 4. The reduction potential of manganese 4 to 3 is greater than that of oxygen to water. So, in its oxidized state, the OEC can split water. This reduces the manganese 4, regenerating the manganese 3 in the cluster and releasing an oxygen molecule. As the light intensity increases, the rate of photosynthesis will also increase, and in a manner similar to Michaelis-Menten kinetics, the rate of increase will begin to level off as all of the photosystems become active. However, instead of asymptotically approaching a Vmax rate, above a certain light intensity, the rate of photosynthesis will begin to decrease until it completely shuts down. This is because as the light intensity becomes greater, it will begin to blow out the photosystems, temporarily deactivating them until all of them are inactivated. This is called photoinactivation and results in a photosynthesis irradiance or PI curve. The energized electrons are transported to cytochrome oxidase enzymes by quinones, which pump protons into the thylakoid space against the concentration gradient. After pumping the protons, the electrons have a lot less of their energy. To be re-energized, the electrons enter the photosystem I complex, which lacks an OEC, and instead uses light to strike the electrons. These then go on to an NADPH dehydrogenase enzyme, where they reduce an NADP to NADPH. The NADPH then goes to the Calvin cycle, i.e. the dark reactions, to reduce carbon. The utilization of the Calvin cycle in oxygenic photosynthesis makes rubisco, the enzyme which binds carbon dioxide to RUBP, the most abundant enzyme on the planet. The ions pumped into the thylakoid space are used to drive the ATP synthase enzyme, and the ATP regenerated is used to both power the cell's metabolism and drive carbon fixation in the Calvin cycle. Apart from being used for carbon fixation, cyanobacteria can harvest light solely to drive their energy metabolism. To do this, photosystem I will use the light energy to energize electrons, but instead of reducing NADP, they'll just be used to pump protons. Once the electrons have lost their energy, they will go back to the photosystem I complex. Here, they are struck by another photon, and in this re-energized state, go back to the cytochrome oxidase enzymes, incrementally lose their energy, and the cycle repeats. This is known as cyclic electron flow. Cyanobacteria uptake most of their nitrogen and sulfur as nitrate and sulfate and utilize the aforementioned assimilatory reductive pathways to include them in their biomass. High ratios of phosphate to nitrate usually favor cyanobacteria over other types of algae. This is because many OTUs of cyanobacteria are capable of taking nitrogen out of the atmosphere to be incorporated into their biomass. As discussed earlier, this is an energetically expensive process. Now, being able to harvest light gives cyanobacteria access to large quantities of energy, but with high levels of phosphorus available, cyanobacteria can produce a much larger ATP-ADP pool. This allows the light energy, which has been turned into a cross-membrane gradient potential, to be more readily shuttled to the nitrogenous enzymes as ATP, leading to a greater rate of nitrogen assimilation. Because nitrogenase is inhibited by oxygen, cyanobacteria which utilize it grow in filaments. In a few of the cells known as heterocysts, no oxygen-producing photosynthesis occurs, so here, atmospheric nitrogen fixation takes place. In marine systems, most of the inorganic carbon is in the form of bicarbonate. As a result, marine algae possess an enzyme called carbonic anhydrase, which converts it into carbon dioxide to be fed into the Calvin cycle. Plants lack this enzyme, and as a result, aquatic and marine plants like seagrass have to uptake carbon as dissolved carbon dioxide. Most marine primary productivity occurs near the coast due to the input of nutrients from the shore. Meanwhile, the open oceans see very little primary productivity because these are very oligotrophic. However, there are areas known as high nutrient, low chlorophyll, or HNLC regions. These have high concentrations of nitrate and phosphate, but very little primary productivity, and include the Bering Sea and Southern Ocean. It's believed that the low concentration of iron in the water is what limits primary productivity in these regions. The sudden release of nitrates and phosphates into the oceans can cause algae to rapidly proliferate. These can be harmful to marine life in one of two ways. First, they can produce toxic compounds and release them into the water. Second, once the algae consume all the nutrients, they die in mass. When this happens, aerobic microorganisms will completely consume the oxygen in the water column as they decompose the algae. This results in anoxia and leads to the formation of dead zones, where marine animals suffocate. Agricultural runoff has led to the formation of dead zones in places like the Mississippi River Delta and the Baltic Sea. While the vast majority of photosynthesis which occurs on the Earth uses water as the electron donor, a few prokaryotes use other electron donors. This is called anoxygenic photosynthesis, and the most common form, photothioatrophy, uses sulfite as the electron donor for the photoreduction of carbon. The two most common varieties of bacteria which conduct photothioatrophy are purple and green sulfur bacteria. Whereas oxygenic phototrophs have two kinds of photosystems and oxidize the electron donor in the photosystem itself, and oxygenic phototrophs have one variety of photosystem, and the electron donor is oxidized elsewhere in the cell, and the electron is delivered to the photosystem where it's energized by the light. Like oxygenic phototrophs, photothiotrophs use the cytochrome oxidase enzymes to pump protons against a cross-membrane concentration gradient to drive ATP regeneration, and use a similar porphyrin-based light-harvesting pigment known as bacterial chlorophyll. Purple sulfur bacteria are part of the incredibly diverse gamma proteobacteria clade and use the SQR pathway employed by other sulfur-oxidizing microbes to oxidize sulfide. These organisms use quinones to move electrons through their membrane, and the electron carrier which delivers it to the cytoplasmic carbon fixation reaction is NADPH. Purple sulfur bacteria also use the Calvin cycle to fix carbon, and their photosystem can be run linearly or cyclically. One environment where purple sulfur bacteria are very prevalent is in meromictic lakes. These lakes are usually very deep compared to how large they are, and as such, they never get completely mixed by wind. This results in the formation of the upper layer, or mixolimnion, which is mixed by the wind, and the lower monomolimnion, which isn't and doesn't interact with the surface. The lower monomolimnion is constantly anoxic and usually usinic. The sulfides inhibit oxygenic phototrophs, allowing purple sulfur bacteria to dominate primary productivity in the lower monomolimnion. In these systems, such as in the Green Lake in upstate New York, purple sulfur bacteria form magenta-colored clouds just beneath the chemocline dividing the two mixing layers. The other main variety of photothiotrope, green sulfur bacteria, can photosynthesize with lower ambient light levels than purple sulfur bacteria, but are more inhibited by any oxygen in the environment. Like sulfur-reducing microbes, they use menaquinones to move electrons through their membrane, and like the photosystem II complex of oxygenic phototrophs, their photosystem can only run electrons linearly. Different OTUs of green sulfur bacteria are known to use the SOX, SQR, and reverse DSR pathway to oxidize sulfur. To assimilate carbon, these microbes use the reverse TCA cycle. Here, the electrons from the sulfides, which were energized by the photosystem, reduce oxidized ferredoxin to its reduced form and NADP to NADPH. These then deliver the electrons to the TCA cycle, causing it to run reductively, binding carbon dioxide to succinyl-CoA and alpha-ketoglutarate to form citrate. The citrate is then reacted with coenzyme A to form acetyl-CoA and oxaloacetate. The acetyl-CoA is fed into biosynthesis pathways, while the oxaloacetate is used for the next turn of the cycle. Although most green sulfur bacteria use sulfur as the electron donor, a select few known as photoferrotrophs use ferrous iron. These inhabit ferruginous lakes and use an iron oxidase enzyme to take the electron from iron II, producing rust. The phototrophic organisms discussed thus far use light to both power their cellular metabolism and produce organic carbon from carbon dioxide, but one variety, known as photoheterotrophs, uptake organic carbon from the environment to be assimilated into their biomass while using light to power their cellular metabolism. Some photoheterotrophs use bacterial chlorophyll in photosystems to drive cyclic electron flow. Others use lipids known as retinols. Similar compounds are used in the light-sensitive cells in our eyes, and, in photoheterotrophs, they are stored in a protein called bacteriorhodopsin. The retinol in the bacteriorhodopsin will absorb a photon, which will then bump a proton off it, causing it to become negatively charged. The proton goes out into the extracellular environment, while a proton from the cytoplasm binds to the retinol. Another photon strikes it and the cycle repeats. This is how these organisms generate a cross-membrane pH gradient to drive the ATP synthase enzyme. Photoheterotrophs which use retinols are common in saline lakes, making them halophiles, and when they bloom, they can make the water turn pink. So that's the metabolic diversity of microorganisms in a nutshell. This is a bit of a broad generalization, and there are far more nuances. What all organisms need is material to construct biomass out of, and energy to enable that construction. Autotrophs get carbon in an inorganic form, whereas heterotrophs get it as organic molecules. Organotrophs drive their energy metabolism by oxidizing or breaking down organic molecules. Lithotrophs get their energy from oxidizing inorganic molecules, and both of these metabolic types are chemotrophs. Instead of using chemical energy to drive their metabolism, phototrophs get their energy from sunlight. The two most prominent metabolic pathways are oxygen-based photoautotrophy and chemoheterotrophy, but many microbes employ different combinations of carbon assimilation pathways and energy metabolisms. Furthermore, metabolically flexible organisms can employ different dissimilatory processes depending on what the external conditions are. The redox ladder often manifests itself in sediments, with aerobic bacteria consuming oxygen from the atmosphere near the surface until it's used up. Going down, once the oxygen is depleted, there's a layer where denitrification dominates remineralization, and once the nitrate is depleted, sulfate reduction dominates. Below that, you're out of electron acceptors, and the breaking down of biomolecules into smaller organic ones, or fermentation, drives energy metabolism. At the interface of each layer, you'll have lithotrophic organisms using the electron donor from the layer above to oxidize the electron acceptor from the layer below, replenishing it. There will usually be a eugenic layer, but its depth depends on the local water chemistry, with it being much deeper in marine sediments than freshwater ones. There may or may not be an additional ferruginous layer, dominated by iron or even manganese respiration, depending on the local geochemistry. A denitrification layer is nearly ubiquitous due to the prominence of nitrogen in biomolecules, but the advent of ammonia and nitrate-based fertilizer has caused more variability in its depth between different environments. One can see this redox stratification in the famous Winogradsky column. In this video, I tried to illustrate how small-scale metabolic processes are responsible for large-scale biogeochemical cycles. Oftentimes we view these as being distinct, but at the end of the day, they are part of the same system. In the next major video, we'll see how many of these systems came into being on the early Earth billions of years ago. If you liked what you see, please like and subscribe because it helps the algorithm out a lot. Thanks.